what I basically did is, and I can give you a bunch of other slide decks if you want. I just put together a bunch of materials from our collection that have a lot of the different topics and tried to focus on all of the different applications that your instrument gives to the university, so, or to the organization. So great.
Okay, sounds good. Yeah. All right. Okay, so I think we have still a few minutes. So
okay, great.
Three minutes to wait. Okay.
How's your instrument performing?
I think it's fine. There was, I think, issue with the HPLC, I think, but I think Shanghai was working on that.
Okay, good. Very good.
Yeah, we started to do some analysis with FAME.
Excellent.
We're still not sure about FAME, but anyway.
Oh, is the FAME still giving you some issues?
Not so much, but I think we kind of.
Just let me know. Let me know, and I can help you out. Yeah.
Maybe take it out and compare, and then put it back.
Yeah, I would go back to without FAMEs and make sure it's performing.
Yeah, sure. Yeah, that's fine.
Okay. Does this look okay from your end?
Yeah, looks good. Yeah. Let's wait.
Yeah, can wait a couple minutes for people.
Yeah. Let people join. Yeah.
give it maybe one more minute and then we can get started.
Sounds good to me. How many are you expecting?
We had 20 to 25 register.
Yeah, it's like YouTube podcast. We can see still people coming in, getting in.
Fashionably late.
Okay, I think we can get started now. Good afternoon,
Sure.
everyone. Thank you so much for joining us. This is the DCCFAR Basic Sciences Core Howard University Virtual Open House. My name is Kara Thompson. I am the Basic Sciences Core Manager. So today, as you can see on the agenda, we'll have a brief welcome and introduction from Dr. Sergey Nikai. Then we will have a presentation from Christopher Bolcato, who is the Thermo Fisher Scientific Specialist we have with us today. If time allows, we will have a presentation from Dr. Tatiana Amasova, who will discuss registration and sample preparation. And then we will close out at 1 p.m. Eastern. So with that, I will pass it over to you, Dr. Nikai.
Yes, thank you so much, Kara. Welcome, everyone. Thank you for joining this seminar. So it's part of our annual series of the open houses. Unfortunately, it's in virtual format because of the COVID pandemic. So this current virtual house will feature our new mass spectrometer. And I would like to take time and thank you, all of you who participated in this, in the acquisition of instrument, providing letters of support and sharing your information and your research with us. So that's very important for us. I think it was super important to use DCCFAR community for the acquisition of this instrument. So, and again, DCCFAR is the inter-institutional program, which basically unites all the universities at the Washington, D.C. area. And the purpose of the basic science core is to provide support in terms of facility use and then facilitating studies in HIV. So, so that's a seminar series. I think that's one way how we can expand the usage of the instruments, and then how we can introduce you as a community users for the all the instruments within DCCFAR. Okay, I think with this, I'm going to give floor to Chris Bolcato, who is the Thermo specialist, and he'll talk about the new instrument that we just received and installed here at Howard. Thank you.
Great. Thank you, Sergey. And thank you, everybody, for taking time out of your day to participate. I'd like to just quickly introduce myself. I'm Christopher Bolcato. I'm one of the application scientists for Thermo Fisher Scientific. I represent the North America omics markets. My role is to visit laboratories and train them on their new instruments, as well as to help with applications. And I did visit Sergey's lab and do some training for Zhang Hao, who I see in the audience there. Zhang Hao, how are you? But they have a wonderful piece of equipment in their lab, and a great operator who's running the instrument now. But what I wanted to do was give you an overview of the instrument and some of the capabilities that it has. And a lot of these capabilities are things that I worked with Zhang Hao and Sergey to set up in the laboratory. So without further ado, let's jump right in. So let's talk in terms of some of the challenges that we face in life sciences mass spectrometry. You'll see three topics here. And these are typically the main applications and workflows that we see in the laboratory that we use these instruments to work through. First and foremost is qualitative proteomics. And one of the challenges associated with qualitative proteomics is we have a breadth of type of samples, which I'm going to talk about in a little more detail, as well as doing protein identifications. We want to characterize those proteins, for example, for determining post-translational modifications. And often the insufficient depth of analysis is one of the problems that plagues us in terms of why we need to have sophisticated instrumentation. Quantitative.
I'm sorry to interrupt. We can't see your screen.
Oh, you can't see my screen. Okay, let's try it again. Let me try to share my screen again. So I'm going to go back. I don't know what happened here. So I'm going to get my presentation and then I'm going to share my screen again. Hang on 1 second. All right, can you see my screen now?
Perfect, thank you.
Okay, are you seeing, as I go back over, are you seeing it advance?
Yes.
Okay, great. So let's just go back from the beginning here. Okay, so what I want to talk about are the type of challenges that we have in life sciences mass spectrometry. And first and foremost, let's discuss some of the application-specific families that these instruments typically service. Qualitative proteomics, which is essentially the understanding of what proteins we have in a given sample, the characterization of those proteins, of not only who they are, what families they come from, but as far as some of the structural information such as post-translational modifications. And one of the issues we've always had with any type of mass spectrometry device, going back in decades, is to maintain a sufficient depth of analysis. So having an instrument that can cover a wide variety of proteins for a wide variety of samples is typically what we are interested in. Quantitative proteomics. Now that once you understand what proteins are found in a given sample or set of samples, being able to accurately quantitate those proteins across a proteome very accurately and with high throughput is definitely one of the big challenges. One thing that I'm not going to talk about in detail, but these instruments are highly utilized for this purpose, are for the characterization of biopharmaceuticals. Protein drug structure elucidation and purity identification. We service plenty of customers who are doing this this application. I'm not certain that's something that you're going to be doing in your core or at Howard, but it's definitely something that this instrument can do. And again, so for these challenges, we have come up with a series of instrumentation that allow this. But right now, at the top of one of the instruments at the top of the line, which is what is housed in Sergey's lab, is the Thermo Scientific Orbitrap Exploris 480 Mass Spectrometer, which is what I refer to as one of our Cadillac flagship instruments at this point in time. It is a next generation mass spectrometer. With the convenience of the multiple sources, whether a high flow source or a nano flow source, combined with the Thermo Scientific Orbitrap Mass Analyzer. They are all in the family of the next generation instruments, which include our tribrid mass spectrometers, as well as our TSQ triple stage quadrupole mass spectrometers. Record-setting performance in their gains in protein or proteome characterization, quantitative workflows, which I'm going to talk about, such as our TMT and SureQuant targeted analysis workflows. And they're engineered essentially to be highly reliable and very robust. And I can attest to that as an operator and as a trainer for these instruments, that they are very, very high throughput and very, very rugged. If you know anything about the instruments that our laboratories produce or our company produces, perhaps you're familiar with the Q Exactive platform instruments, which was our flagship in the past decade and a half. The Orbitrap Exploris. Let me get my little pointer out here. The Orbitrap Exploris, which is on this side, the 480 Orbitrap Exploris 480, is much smaller than its predecessor, the Q Exactive. You can see the footprint in the bench is much smaller than what we were looking at before. So it offers a lot more punch in a smaller size. And as far as capabilities, I'm just going to go through these very briefly. This is sort of the portfolio of Q Exactive instruments. And as we go up in price, obviously we go up in performance. But what the Orbitrap Exploris offers are better capabilities than even the top of the line Q Exactive HF, which is the last in the line of the Q Exactive instruments. And I'm going to call out some of the functionalities. One being in particular, the resolution ability of this instrument is much higher, is actually twice the highest capacity, which is the resolution of the mass spectrometer is the ability to determine between two very similar masses in the instrument. So we have the ability to go and determine exact masses, which much better accuracy and mass resolution in this instrument. So this is more of a marketing type slide. This shows you some of the specs for the Orbitrap Exploris 480. We have a mass range of up to 6,000 mass by charge. There's an optional upgrade for a high mass option to go up to 8,000. So you can imagine we have a very wide mass range detectable by the instrument. We can go down to 4.4 units, 0.4 mass to charge units, so we can get exquisite isolation width in our quadrupoles. And the scan rate is up to 40 hertz for MS and MSMS, which allows us to go deeper and deeper and to give you a more wide, diverse range of identifications at a very high rate, as well as the high resolution, which I discussed before. The dynamic range is approximately 5,000 to 1, which allows us to identify and even quantify over a very wide dynamic range. We have a variety of different ways that you can dissociate or fragment ions, but in this instrument in particular, we have the higher energy collisional dissociation, which we're going to be doing for MSMS characterization as well as quantification. When you take the hood off the instrument and look under the cover, this is essentially a schematic of what we see in the instrument. Starting here out at the front end of the instrument with the Optimax next generation electrospray ion source. This is where we would actually generate our ions in the source of the instrument, no matter whether we're doing high flow or low flow. So as far as we go into the instrument, the Optimax ion source with its high capacity transfer tube allows more ions to push into the mass spec, and it allows improved sensitivity. It efficiently captures ions and pushes them through. One of the new developments in this type of instrument, because we have a higher capacity source, we need to funnel those ions in. So we have an electrodynamic ion funnel, essentially to harvest and pull the ions into the source. We have an advanced active beam guide, which essentially helps to increase the robustness of our instrument by removing the neutrals as they move through. We also, I hesitated to mention also that the easy, reliable IC or internal calibrant source, which enhances our ability to for real-time calibration on the fly to ensure that our accuracy of our instrument is spot on. And this is something that I recommend customers work with when they're working with their instrument. It can ensure, especially in this situation where they have fluctuations in temperature or regional variations in their laboratories due to different locations, exposure to different elemental things. The IC can help to maintain accuracy of the instrument. The advanced active beam guide increases our robustness by removing neutrals. Essentially, the ions would have a charge will make this bend here, but neutrally charged ions will actually crash into the metal on each side. So each instrument has a variation of this. This is essentially what you have in your 480. We have the quadrupole mass filter, which allows us to allow specific mass ranges into the instrument. So when we're doing our full scans or our data-dependent scans, we can essentially customize what masses get through. So this helps us to do a variety of different experiment types, and we can go down to very, very narrow widths to ensure that we're getting the appropriate ions isolated through the quadrupole. We also have, in this type of quad, we have a segmented design, which enhances our ability to isolate specific mass ranges and transmit them efficiently through. We have our ultra high field Orbitrap mass analyzer, which I talked about before, which offers resolution up to 480,000 and scan speeds up to 40 hertz, which allows us to do some of those experiments with exquisite timing. Our automatic gain control is assisted by an independent charge detector, which ensures that our instrument fills with the appropriate number of ions per scan. Again, I'm going into some high advanced details about the instrument, but I wanted to talk about some of the key components. We also have our ion routing multipole at the back end of the instrument, which serves as a bucket for which we can fill our ions with or do our fragmentation types. If we look at the Orbitrap itself, and we have a picture here of a couple of different type of Orbitrap instruments, or Orbitraps, we have the ability to look at different sizes of Orbitraps, and the different sizes of Orbitraps have different capabilities as far as their resolution. We have the high field Orbitraps, and we have the lower field Orbitraps, which are associated with our older instruments. So the smaller ultra high field Orbitrap is what you have inside of your mass spectrometer. And again, they're very simple. When you look at them, they're a couple pieces of metal, some ceramic. You have an inner spindle and a couple outer electrodes, but these are really the heart and the soul of the mass spec. And essentially, the Orbitraps work by spinning ions inside of this orbital trapping device, and it's this rotation, this frequency of our axial oscillation and the rotation around that Orbitrap, which give us the ability to determine what the masses are based on the frequencies and a constant that we establish when we calibrate the instrument. Essentially, the Orbitrap works by taking a short packet of ions and pulling them into the center of the Orbitrap, and then we change the potential around the central spindle, and then those ions begin to spin. I apologize that it's forming a not quite perfect oscillation around, but essentially the ions should be spinning around the center electrode of the Orbitrap. And then when we remove that voltage, the ions essentially squeeze and enter a stable trajectory around the Orbitrap. And it's that rotating ring which allows us to characterize the frequencies for specific masses to charge ions. So when ions enter the Orbitrap, they will, once they are stabilized, they will rotate or oscillate back and forth in these stable rings, and essentially those efficiencies, or I'm sorry, those frequencies are then determined based on the image current that is imparted upon these outer electrodes. And it's these frequencies that we measure. So what we're looking at here is a complex frequency of multiple frequencies overlaid. So it's kind of like it's a complex frequency called a transient. And if you take those frequencies and do a Fourier transform, therefore you can go further and isolate individual frequencies in order to determine what those masses are. So we take our raw transient, which our Orbitrap measures, and then we use a Fourier transform to isolate the individual transients as well as their amplitudes or their heights, and then take that data, reduce it further, and get this frequency intensity distribution, which is starting to look more like a mass spectrum. And then we use the Orbitrap, the calibrated constants around the Orbitrap, take these measured frequencies and back calculate in order to determine our mass spectrum. So this is what the instrument does in the background. Along with a highly efficient mass spectrometer, we also need to couple these with a highly efficient LC system. And one of the things that Angelita wanted me to touch upon is the fact that when we go to lower and lower flow rates, and what we're looking at is the column inner diameter here, as we're going to more tight and more narrow column IDs, we're actually improving our sensitivity for detection of ions by concentrating our flow and concentrating the ions into smaller and smaller packets. So when we go from high flow to low flow, what we will see is a great intensity in our ability to have better sensitivity. So when we're doing proteomics experiments, what we want to do is push towards the nano flow rates, which you can do on the RSLC nano instrument that you have in the laboratory. So we'll get improved ionization efficiency with smaller droplets, increased surface to volume ratio, and essentially what we will get is an increase in our analyte concentration with a decreased column ID. This is just a image of what you have in the laboratory. You have the RSLC Nano Ultimate 3000, which allows us to do some really low flow experiments. We have the nano pumps housed up here. We have a separate loading pump. We have a column chamber where we can do trap elute experiments, which we have set up in Sergey's laboratory, and we have a cooled auto sampler. All of this together allows us to do those low flow experiments with ease of trapping. I'm going to cut forward a bit here to move. Let's get into the meat of the talk, which essentially is talking about how we are going to use the Orbitrap Exploris 480 in order to do some of these experiments. So let's talk a little bit about qualitative proteomics. So what are we doing in qualitative proteomics? Essentially, we were trying to identify what proteins are found in a typical sample. And these samples can be very simple to very complicated samples. We're also trying to get an idea of characterizing what post-translational modifications may be on the proteins that we identify. You know, are these phosphorylated? Are these ubiquitinated? Do they have things that can provide some information as to what's going on in our biological system? As well as I categorize qualitative proteomics as with the application of label-free quantitation, which is essentially getting an idea of relative differences sample to sample. And this can be very useful in looking at disease versus control populations, for example, or drug-treated samples versus non-treated. So typical samples, and I worked in a core facility at the University of Pittsburgh for a number of years. The typical samples that we would get on a day-to-day basis are essentially our in-gel samples. So in-gel digestions of 1D and 2D SDS page. People come with their gel bands or gel spots and want to get a feel for what proteins are they identifying. Very typical sample prep. We also get the in-solution digestions where they're taking some sort of, for example, a biofluid, blood, plasma, urine, cerebrospinal fluid, perhaps with or without a high abundance protein depletion to get rid of albumin or immunoglobulins, and then taking those samples and digesting them in solution or doing some sort of mud pit or strong cation exchange fractionation or high pH fractionation. So either something very simple or something very, very complex, like a biofluid, or a tissue homogenate, cell lysates, or even now we have the capability to do single cell preparations for single cell proteomics. And I want to call that out as being one of the newest advances, and I have a few slides to share on it. The hallmark, and I would say bread and butter type experiment, is the data-dependent peptide protein identification, which we do on the Orbitrap Exploris. And this is essentially an example of a typical application where we're doing, we have the benefit of extraordinary sequencing depth and figuring out what proteins are present down to low-level nanogram amounts of digest injected on column. And here we see in these two diagrams, we see the number of proteins identified and the number of unique peptides identified by doing experiments with typical loads of 50, 100, 200, 500, and 1,000 nanograms on column. And this is what you would typically, depending on how much material you have to work with, but this is what you would typically see from the number of proteins identified. And as we go up with the amount injected, as we look at the average or the sum of 3 individual replicates injected on column, we see that we're increasing the number of protein IDs as well as the unique peptide IDs from low amounts to high amounts. Now, 50 nanograms is certainly not the limit. This is just an example from a poster we had at ASMS 2019. And this is a typical situation where we would run samples and do a full scan, so doing a data-dependent assessment of the samples, doing a 60,000 resolution or 120,000 resolution for full scan to set up our peptide identifications at the intact peptide level, and then from those full scan experiments, deciding on an automated manner to do data-dependent MS2 on the peptides that elute, and then getting some structural information. And in this situation, this is typically what I would set up: a top N or a looped experiment where we take the top 20 most intense ions and identify those in those experiments. And this is using a 50 centimeter nano column with a simple 60-minute gradient. Moving forward, doing an example where we're doing label-free quantification. This is an example from the same poster done on the Exploris 480, where we are creating an artificial situation where we have taken human HeLa cell lysates and spiked them with increasing amounts of a yeast digest, where we know the expected ratios. So, for example, if we look at the highest amount loaded, 250 nanogram versus 125 nanogram, that's essentially a 2-to-1 ratio, a 5-to-1 ratio, and a 10-to-1 ratio. By creating these artificially, quote unquote, stack the deck in our favor experiments, we know the expected ratios, and we can perform these label-free quantification experiments and essentially recapitulate in the data, showing those expected ratios of the proteins identified across those samples. And actually, our expected ratios are actually quantified with good accuracy in these samples. So we can look at samples where in this case, we're looking at samples where we know what the expected ratios are, but just imagine what you can do in samples where these are real life samples where you are looking for fold differences between groups. Moving forward. On top of having an exquisite mass analyzer and capabilities, we also have a FAMES Pro interface, which helps us to enhance our selectivity by removing, using the FAMES, which is a frequency-assisted ion mobility mass spectrometry source on the front end, which allows us to remove selectively background ions, which boosts our sensitivity. And essentially, this is a cutaway of what the ion mobility source looks like. And essentially, what we're doing is we're adding let me go a little bit further. We're ramping voltages up and down in a manner that allows us to cause ions to shift in their movement through the ion source. What that does for us is it allows us, as we ramp these voltages up and down, it allows us to remove specific charge state ions. And what we can do is we can customize these compensation voltages in order to remove singly charged ions from the background. And where do we have singly charged ions? Pretty much for any type of sample. They are contaminant ions, which if we can remove those, we can enhance the selectivity for triptych digest peptides, such as the doubly, triply, and quadruply charged ones we see here. And if you look at it, there's a quick cartoon of this, we can see that as we're removing these ions at the ion source, it's allowing our triptych peptides to enter into the mass spectrometer. So we can do a sort of mass spec filtration of ions to remove those to enhance our sensitivity. And what we're seeing here, an example, is an example of doing the same type of experiment with a HeLa digest on a 120-minute gradient with the FAMES Pro interface. And we're able to see very good reproducibility between these two samples, between these 5 injections, for example, and maximum coverage and reproducibility of 6,700 proteins identified in a 2-hour run with 200 nanograms of HeLa digest on column. So the FAMES Pro, what we do with this FAMES Pro is we cycle between different collision or compensation voltages in order to isolate and remove interfering ions in order to boost our signal for the proteins that we're trying to identify. And these are just some examples. That was 200 nanograms. This is an example down at 10 nanograms of HeLa loaded on column with a 90-minute gradient with the FAMES Pro interface. They were able to identify almost 3,000 proteins in 90 minutes with down to 10 nanograms of HeLa digest. So think in terms of your precious samples. If you only have several nanograms of digest to load on column, for example, using the FAMES source, you could enhance the sensitivity for identifications. So this is one upside to using it, and it's quite reproducible. Similarly, you can run the instrument with different gradients, and the only point I wanted to show here is running longer gradients. Again, you can save a little bit of time by shortening it down to a 90-minute gradient or even a 60-minute gradient, and you're going to get similar results to longer gradients. So it has excellent performance over a wide range of gradients, allowing you to separate out peptides and identify proteins. And we can increase our capabilities by looping those FAMES voltages throughout the course of the run. This is an example of label-free quantification of using a similar profile, using a HeLa digest spiked with various amounts of E. coli. So they took E. coli protein digest and spiked into a constant background of HeLa digest to basically provide a background material to obtain a ratio of approximately fourfold. They then analyzed these on an Orbitrap Exploris 480 with the FAMES Pro interface. Very, very simple, similar experiments as before, except we're doing a simple 60-minute gradient of these 3 replicates over 4 different concentrations and using a single FAMES voltage. And then this data was run through the instrument. I just wanted to call out that in building methods, and this is something I went through with Zhonghao in great detail, a lot of plug and play type methods, and we as application scientists will walk the users through this. These templates are already built for the user, but we essentially will guide them through what the important details of them are. Base peak chromatograms for the 25 nanograms of E. coli compared to the 0.625 nanograms of E. coli. These are the basically base peak chromatograms, showing a very good distribution of peptides. They're almost superimposable, so you're looking at similar distributions of peptides across these 2 runs. When analyzing this data, we use Proteome Discoverer 2.4 with a processing and consensus workflows, which are essentially ready to go, plug and play for the user. They just wanted to show you what sort of the analysis workflow looks like for Proteome Discoverer, which is our protein identification and quantification software. If we look at Venn diagrams, which can export from Proteome Discoverer, for these injections of the 200 nanograms of HeLa spiked into E. coli, spiked with 100 nanograms of E. coli. If we look at the E. coli proteins, we have good overlapping confidence in comparison of the peptide groups as well as our protein groups. So the proteins identified, we have them across 3 runs. We have 85% agreement across the proteins identified. You're going to get a little bit of variation from run to run, but within almost 85% of those proteins are covered by 3 replicate analyses. So we have great matching across multiple runs. And as we look at the overall proteins identified. We've got a large number of proteins identified in both the E. coli and the human at the 2 different ratios. But what I really want to call out is if we look at the precision and accuracy of the proteins identified, we're getting a good agreement in, and this is on a log2 ratio scale here. We're looking at a 4-fold, a 4-to-1, 4-to-1 estimation of relative protein amounts between these samples. Again, we loaded E. coli. We spiked the deck in the favor of seeing a 4-to-1 ratio here and a 4-to-1 ratio here, with the humans being human proteins being 1-to-1. So we're getting excellent precision and accuracy in our quantified applications. From the standpoint of doing protein quantification or protein qualification, protein identifications, it gives you essentially what I would call a phone book of understanding what proteins are present in a given sample. Along with that, we can get an idea of what. Oh my goodness, did I lose you? Please tell me I didn't lose you.
No, you're fine.
Okay, for some reason I thought that I closed the Zoom window by accident. My apologies.
You're good.
Your face has all disappeared and I just freaked out for a minute. Okay, so let me go back for a second. When I'm talking in terms of qualitative proteomics, we get an idea of what proteins may be present in a sample and relative differences between samples, which is great information. But what if we want to take it further and get a better accuracy in our protein quantitation and do things in higher throughput? Okay, so we're talking about the same type of experiments or same type of samples as before: tissues, biofluids, cell lysates, or single cell preparations. But now we can stack the deck a little bit further and start talking in terms of our TMT workflows. And this is something that the lab at Howard has the ability to do. I'm going to talk a little bit more detail about what these tandem mass tag experiments can do. This is still another relative quantitation type run, but the beauty of it is that we can multiplex and compare from 2 up to 18 samples in one analytical run, which is a wonderful time saver and ability for the ability for multiplexing. We also have absolute protein quantification workflows, such as our hallmark parallel reaction monitoring with heavy labeled peptides, or our SURE workflows. And I'm going to try to get to this in this presentation, but due to time, we might not get through all of that. I'm going to try to run another 15 minutes and leave some time for questions. Very briefly, we want to be able to, as well as identify, we want to be able to quantify proteins with high precision and accuracy. And with using the TMT, we can quantify thousands of proteins simultaneously in one run. We also want to make sure that we are doing the appropriate method for quantification. We want as precise as possible in using our instruments. And the Orbitrap is going to give us, depending on what type of method, whether it's an SRM, a multiplex, or a label-free quant or DIA, we're going to have differences in how well we can shoot the target. But the big thing is that in Orbitrap systems, we have good mass accuracy, but might not always be right on the target.
Chris, I'm
Yes.
sorry to interrupt again, but you have about 5 more minutes.
No problem. I'm going to go as fast as I can.
Thank you.
With the isobaric tags, we have the ability to multiplex. And when we label our peptides, we label them with reagents which essentially bind to the peptide, and then we can cleave them to not only do fragmentation for identification of the peptide, but we can release these reporter ions so that we know and get an idea of the relative quantification from each sample. Using high resolution, we're able to pull apart these isobaric tags. And they come in a variety of flavors. This is, for example, an 11-plex. So we could multiplex up to 11 samples using those different tandem mass tags. So we could simultaneously quantify as well as identify proteins in those experiments. And this is just the TMT 11-plex yeast standard, which we use for training purposes. And doing TMT, we're able to, again, with different applications, the FAMES being the most superior, we're going to be able to quantify as well as identify with good accuracy a number of proteins across a given sample. In Proteome Discoverer, then we can look at these different relative differences. We can use volcano plots to look at our outliers and to find out what is statistically significant between groups. Here we would see the down-regulated as well as the up-regulated proteins. We're able to do principal component analyses, as well as clustering for using heat maps. In Proteome Discoverer, we're able to do very specific TMT methods, depending on what type of plex we're using. Automated workflows for laying everything out in our processing and consensus workflows, creating ratios for study factors, using replicates, and looking at the data. And this is just an example of that triple knockout. We see differences upon a principal comparison and principal comparison using the triple knockout, as well as seeing the true knockdown approach. So this triple knockout has a parental strain versus 3 metabolic knockouts. And here you can see this MET6 gene is truly knocked out versus the parental here. And we can see in the volcano plots that other proteins are tracking as they upregulate and downregulate. We also, I'm going to skip through this, but this is our SureQuant application workflow, which you can look at in the slides on your own, since we're going to run out of time. But the SureQuant targeted protein quantitation essentially are kits which allow you to quantitate a number of proteins in a given sample. They come in the AKT-mTOR regulatory pathway, where we can look at up to like 26 proteins. These are heavy labeled peptides you would essentially spike into your sample. I'm gonna rip through that quickly because I do want to talk quickly about single cell applications, which is the last thing I want to talk about. We talked about different sample types, cells, in solution digestions, but what if we wanted to look at specific populations or subpopulations of samples? We have the ability to isolate specific proteins from a cell prep, and then isolate those, and then you do the proteomics or protein relative quantitation on a limited amount of sample. So this is a new application. I did train the laboratory to do approach the single cell proteomics type applications. There's a lot of literature out there that talks about different preparation methodologies. We're able to do low-level loads using the Exploris, and essentially these low-level loads, what's beautiful about it is on these low-level loads we can approximate the amount, we can approximate identification and relative quantification of proteins in low-level loads, approximating that of single cells, and actually identify as well as relatively quantify proteins in low-level loads from those type samples. And that's essentially, I know I had to rush there at the end, but I wanted to touch on all the typical applications that your instrument, there's lots more, but the qualitative, quantitative, qualitative as well as relative and absolute quantification, and even down to the sensitivity of single cell proteomics. I'm going to stop there and allow for some questions because we kind of were rushing there at the end. I think we were at about 12:45. Perfect. I rushed so that it gives us about 5 minutes for questions, and then we can get into your sample prep discussion. So why don't we open it up for questions from the group?
Thank you, Chris.
Yeah. No problem.
Okay, so yeah, so we can have people ask questions, or you can put them in the chat as well. So if you have questions.
Okay, so I have a couple in the second. Okay, we're going to talk about the sample process stuff. I'll let you guys answer that, and then what are the new features for Proteome Discoverer 2.5 compared to 2.4? There's a lot of difference. Most of it being that we have the ability to do the short quant applications in 2.5. We have the ProSit spectral library searching, as well as some of the visualization and doing the multiple precursor searching. So you have more modes available to you in 2.5 versus 2.4. Again, they came out pretty close in release times. So there's not that many more advances in 2.5 versus 2.4, but the one big thing with 2.5 is there we no longer have the maintenance licenses available for that maintenance license. So from 2.5 on, once you have it, and you don't have to do that yearly maintenance on top of that. But you could demo it if you were interested in testing it out. But 2.5 is the version that they have in Sergey's lab, which they're going to be doing all of the data analysis for you.
So the users they can download demo version, right?
Yeah, they can download a demo version and practice on it on their own if they would like to. There's a series of familiarization workflows on there. So you could download label-free, TMT, SILAC. There's a bunch of different scenarios where you could download the data and practice with it. Sergey could send you my information if you'd like some informal training on that. And then once the 60-day period expires, you actually have a viewing software. Yeah. So if the lab were to process your data in PD, you could actually open your data up in PD on your own and browse it. You just can't do any searching.
Yeah, I think that's very important. So that's what we try to do with it.
Yeah, exactly.
Well, okay, thanks.
What other questions can I answer? I know we're a little bit short on time, but I'd be happy to answer some questions.
I mean, we can have questions from audience. So if anyone
please,
has
jump in.
Please, yeah.
Type it in or just unmute yourself and ask.
Good. Looks like we're good actually. Let's go to so let's switch to the second part. And
okay. Do I need to stop sharing?
I think you can stop sharing.
Okay.
And then let me see if Tanya, you can share your screen if you have presentation. And I think you can specifically focus on the questions about the how the users can come and what they can do. I think, right? Because I think Chris covered lots of different things in terms of the data.
Oh, absolutely. Mm-hmm. Okay. Excellent talk, Chris. Thank you very much.
No problem. A lot to cover in 45 minutes.
Absolutely. I mean, you cover everything. So it's impressive talk. I just wanted to add a little bit about sample preparation. As for mostly, I mean, you covered basically everything, but I can talk a little bit more about samples as for users, right? What actually users need to do, have to do, like to bring their samples to our facility. Basically, you can bring different kind of samples. You can bring, as Chris said, gel pieces. This would be, would be the best probably solution for everyone. But sometimes you don't know like what exactly you're doing, and sometimes people just bring cell lysates or even like organs and tissues. So like for that, we need to explain a little bit what to expect and what they basically need to do, like a little bit with samples. Like any biological system, of course, you want to have experimental design for your samples, what exactly you want to see, you want to have in the end, because this method is absolutely marvelous and it can show you a lot of results, but if you don't know what you're looking for, all these thousands of proteins can be just hard to handle and hard to analyze. So like, as I said before, sample preparation is very important part of getting your results in the end, because you can't rely only on excellent instrument and software, but you need to think about your sample preparation, because 30% or more of your input, like in your result, will be affected by your design and sample preparation itself. Of course, as again Chris said already, you can use different like tissues, organs, bodily fluid cells. This LCMS gives you a lot of variety of possible samples you can actually use, and you're going for a huge variety of proteins, or you can go and focus on particular post-translational modifications of particular proteins, or identify basically interactions, protein-protein interactions of protein of your interest. It just gives you a lot of possibilities to work with biological systems of your interest. Basically, as for any sample, will be like several steps, starting from lysing your cells or organs or tissues, and basically this very important step will include not just lysis by itself, but also like you need to think about fractionations of your proteins, because if we're talking about focusing on membrane proteins or like nuclear proteins, or like particular proteins which you focus on some excreted proteins. It's just you have to think about like how you what you focus, like what is aim of your research. And then of course it's like digestion. I mean, the bottom up, it's basically we deal not with proteins but with peptides, right? And from peptides we're getting back to proteins. So it means it has its own limitations, because excuse me, because basically not all proteins, of course, can be digested with enzymes, right? If you have highly heavily glycosylated proteins, then you have to basically go like around and deglycosylate them first, and then just, trying to. Some proteins, they just don't have trypsin sites, digestion sites, so we have to go for another enzymes to do digestion. I mean, it's like all these things, they can be pretty complicated, but easy as well from our side. We just need to know, and you need to know what we're dealing with, what proteins of interest, or what is the goal basically of the research. Of course, sample preparations has its challenges, and as again we said before, mentioned before, SDS, I mean, the gel samples, they like best basically for mass spectrometry, and they give a lot of information, and in gel you can get well denatured proteins. So, and basically usually from gel we can get much more data from comparing to straight lysate from solutions, for example. For solutions, I would say would be better like low protein samples. Let's say, for example, like urine or tears, the liquids which basically they don't have too many proteins. And if we're talking about tissue, of course, it should be processed in several steps before it will be going for trypsin digestion. Otherwise, we will get only major proteins, right? As proteins which kind of overwhelm the machine, and in the end we can get much less information about proteins of interest.
Yeah, 2 minutes left for you, Tanya.
Sorry. Oh yeah, I should, yeah, like for example for blood, for plasma, right, it's only less than 10% of proteins basically. You can, they not high abundant proteins, and they can be like all these proteins of interest. And 90% it will be the same proteins. Majority of it will be albumin, of course, immunoglobulins, and so on. So like everyone has to understand that all these abundant proteins, they will be detected by mass spectrometer, but not like low abundant proteins. And so for that, you need to do like one extra step to get rid of abundant proteins. There are many kits for that, or you can use antibodies or some other methods to remove these proteins. So about quantitative, yes, it's already, Chris already talked about TNT. I also want to mention SILAC. It's a method where you can use, you can label in vitro, in vivo, in cell culture, basically. You can label proteins. If in tandem, in TMT, we label peptides, in this case, proteins in vivo. And but basically, majority of people, they use label-free quantification because now, like nowadays, instruments, they so good and software so good that basically it's possible to compare treated, non-treated, let's say, samples and do it without any labels. But also, yeah,
I think you have to jump to the end now. Yeah.
Yes, and in the end, let's say SRM and sure quant is something new. I'm not very familiar with. It's basically a system only for cancer research for now, as I understand. But CRM is good also for systems where you don't have too many proteins, too much of proteins. So again, like for example, like for urine, right? But if you have a complex system, this is hard to work with, let's say. You have to do, again, several steps before we can imply the system. So this basically our old, let's say, we didn't discuss yet our new, price list, but it's going to be approximately close to this one. It means we can do, I mean, we can process samples, let's say, starting from cell lysate, right? Or we can process samples which you already, like you already did several steps before. You know, it's all depends on the users, and if you have any questions, we can provide training, consulting, you know, like just for give you some protocols for particular sample preparations. So please, if you have any particular question.
I think we have to stop here. Thank you, Tanya. Thank you so much. I'm trying to push. So I think she didn't mention, but we are setting up the electronic system for signing up. It's I think it works now for signing up, but it still doesn't work to collect the data on the users. So we're still kind of troubleshooting it. So, but I think it will be up and running very, very soon. So I think if we have maybe last remaining questions, you can ask. Right.
I just wanted to jump in real quick for both of you. If you would like me to go into more detail, you could set up another time. I could go through in detail label-free, TMT, SureQuant. There's a lot of detail to it that's hard to cover within an hour, but you know, you have the system for it, and for folks to understand what they can get out of it. I'd be happy to provide more. I'd be happy to answer any of their questions that come up. Just keep pass my information on to the group, and I'd be happy to help.
Yeah, I think what might be of interest for the users is maybe to get some additional training on the data analysis, because I mean it's not really part of, because we don't have bioinformatician on board, and we can do that, but that actually takes lots of time. And then so if someone is interested, that may be something we can set up.
We could do that because I have data. Yeah, I have lots of data sets, and we could do a 1-hour tutorial on how to load it up in PD and how to view the data and look at it in PD. I'd be happy to do that. What you kind of saw was, perfect scenario.
You're on mute.
Great.
I'm mute. Yeah, so I think we're going to stop here, and I'd like to thank everyone, and Kara, I think it's a wonderful job, and again, thank you everyone for joining our seminar series, and I think we'll have next one in September, hopefully. So, and also I think we're going to have the DCCFAR research symposium in end of August. So please register if you didn't register. So I think we'll be happy to if you join. Thank you so much. Take care.
Thank you for having me. Bye-bye.
Yeah, everyone.